Okay. There we go. Okay. Yeah. So we're kind of just carrying on with kind of general cellular physiology type things, some cellular anatomy. Okay. So we're talking about some very key principles that we're going to continue on through this whole year in this class, which is solutions and diffusion and osmosis. And so some things like that today. So um, the definition of a solution is a homogeneous mixture of components. Okay. So just a bunch of stuff mixed together. Okay. Those components are two things, the solvent and the solute. Okay, the solvent is the water part of that, okay, the liquid part of that, and the solute is the stuff, kind of the little particles floating around in there, okay, whatever substance is dissolved in that solvent. So we put a solvent and a solute together, that's what makes up a solution. Okay, so we've already talked about one of water's properties being that it's a universal solvent, right? That it breaks down most things and dissolves them into a solution. Okay, we already talked about how it breaks down ionically bonded particles or compounds, right? Breaks down polar substances, not so much with the non-polar ones, but breaks those things down. And then we have something that's kind of like a solution, but not quite, called a colloid, which is a heterogeneous mixture, meaning that so with a solution, if we had a cup with a solution in it, and we took a little teaspoon of liquid from that solution from the top, and then we took a teaspoon of liquid from another part of that, they would have relatively the same amount of water and other things dissolved in there, right? So that's the homogeneous part of the mixture, right? But with a colloid, it's heterogeneous, so it's not mixed up perfectly, right? So we took a scoop from one spot and a scoop from another spot. They wouldn't have the exact same components or same relative components in there, okay? So think of that one a little bit more like Jello, okay? Um, and we'll talk about the cell cytoplasm here in a second, but that's the stuff inside of a cell. Um, the cytoplasm is kind of somewhere between these two things, kind of somewhere between a solution and a colloid. Um, Okay, so what kinds of things do we find mixed up in solution? Okay, the first one is lots of salts. Okay, so salts are bound together with an ionic bond, right? We talked about those ionic bonds last time, okay, where we have one atom giving an electron to another one, and now they have opposite charges, now they stick together, right? So sodium chloride was that great example, okay? So all ions are also electrolytes, which means that they can conduct electricity. Okay, so that's something that we'll talk a lot about when we get to when we talk about muscle contraction, and we talk about the nervous system and how signals are sent down neurons. Okay, it's totally dependent on ions, charged particles crossing over a membrane of those cells, and that's really electrical activity in our bodies. Okay, so the properties of salts are that they're crystal when they're in their solid state, so kind of form this crystal structure with sharp, rigid edges, and then they dissolve in water, right? And we talked about last time kind of how they dissolve in water, which is, sorry, um, try to get my pointer here. Can I do a pointer? Get so used to my method of my iPad and now I'm lost. No, I know I can do a pointer somehow. There we go. Okay, yeah, there we go. So we talked about this last time where we've got these polar molecules of H2O, right? The oxygen side's more negative, the hydrogen side's more positive, right? So we take this sodium chloride, all of the negative oxygens go up next to the positive sodiums, right? The positive hydrogens go up next to the negative chloride, and that water's moving around a bunch, right? So it just rips the sodium chloride apart, right? Because it's stuck to those things, and that's how it dissolves it, right? That's how it dissolves salt. Okay, so what makes a particle an ion? It's that it has a charge, right? Positive or negative charge, okay? And then the ionic bonds we've already talked about, right? That well, an electron is transferred from one to the other. Now the one that gained the electron has a negative charge, the one that lost the electron has a positive charge, and now they stick together. And so water's good at dissolving those substances. Okay. All right, so yeah, this is just kind of solidifying that exact concept here. right? So if we take a salt, we take a polar molecule, and we take a non-polar molecule. Um, this is just showing how water is good at dissolving the salt, right? We just looked at that, just went through how that works with the positive parts of water sticking to the negative chloride, right? And then it's the same thing with another polar molecule, like carbon monoxide. Has the oxygen side's negative there. The carbon side is positive. So same thing, the positive part of water sticks to the negative part of oxygen, negative part there sticks there. And again, it just pulls those things apart and dissolves them, right? But when we have a very non-polar molecule like methane, it doesn't do that, and it can't pull it apart. So it just ends up making two separate layers, right? When you put oil in water, it's the same kind of idea, it just makes two layers because it doesn't mix up, doesn't dissolve. Okay, so all of this is kind of leading up to the solution or the stuff we find inside of cells, which is called cytoplasm. Okay, so cytoplasm is the contents of the cell between the cell membrane. So with inside the cell, between the cell membrane and then the nucleus. Okay, so not inside the nucleus, but everywhere else inside that cell is cytoplasm. Okay, so we're going to talk about all the different things that we'll find in there, all the different components of cytoplasm. Okay, so one of those components is cytosol, right? So the cytosol is the solution-like stuff inside. So it's semi-transparent. It's somewhere between a solution and a colloid, like I mentioned. So not quite jello, not quite a homogeneous mixture, but somewhere in the middle there. Okay, and so we have all kinds of dissolved stuff like salts and glucose and oxygen and CO2, all these things that we need to have inside of our cells, as well as ATP, different proteins. Okay, we have the cellular organelles, right? We'll be talking about those more as we move on, but the rough endoplasmic reticulum, the mitochondria, all those different things that are like kind of like little mini cells inside of the cell that have all kinds of functions. And then certain cells have unique parts to them. So those would be the inclusions. So for example, we have two primary places in our body that we store um, carbohydrate in the form of glycogen. Glycogen is just a bunch of glucose molecules strung together. So it's really just in our muscle and our liver that we store a lot of it. And so those would be unique things we find inside of those cells. We wouldn't necessarily see that inside of other cells. And then the cytoskeleton, and we'll talk about the cytoskeleton next, but this is kind of like the internal structure of our cells. Questions on any of this so far? Okay. All right. So the cytoskeleton, we kind of have three primary protein categories that make up our cytoskeleton. And so these are different protein structures that really give the cell its shape, help it move, help it kind of have an internal system of transportation as well. And so we have our actin filaments, which are also known as microfilaments. Okay, these are all made of actin fibers. This is the protein called actin, which is the same one we find inside of our skeletal muscle. We'll talk about later if you've heard of actin and myosin. Those are the ones that help us contract our muscle. Okay, so the functions of this are, number one, give the cell its shape, give the cell its strength, and then allow the cell to move in cases of cells that are able to move. Okay, we've got our intermediate filaments. These are kind of these tough protein fibers that are most stable and permanent. These are kind of like the skeleton of the cell. Think of these as like the bones of the cell. So they're mostly just there providing support. Okay, and then finally our microtubules. These are the biggest, but they're hollow. And these kind of also give the cell some framework as well, but they also serve as like an internal transportation system inside the cell. They serve as these little like train tracks of kind of like moving things around cells. And it's pretty fascinating. Some recent discoveries have kind of shown just how much movement there is inside of cells. You guys have probably seen, some of you have probably seen some of these videos where there's these little things kind of walking on these little tracks. So let me play this one right here, if it works for us. Made a mistake and didn't test the audio before, but let's give it a shot. Okay, what this is just like a little experiment showing kind of a time-lapse video of a plant cell growing. And then once we see the kind of length of this plant cell, we can see things traveling back and forth on these microtubule filaments. So there is kind of cool music. Let me see if I can get that to work real quick. It's not that cool, but it's just better than being completely silent. Look at all those speaker options. So many. Nope, still not the right one. We can listen to it in silence if we need to, but I'm going to try a little bit longer. Just play it on my computer speakers. They're pretty loud. Hope I work. Yeah, we'll just go that way. Okay. Okay, let me get it back right here. Yeah, so that is what's going on in our cells all the time. We have all of these actin filament, or these microtubule filaments all throughout our cells, and we're constantly having to move things around our cell. So we might need a mitochondria in a different spot so those little motor proteins can grab onto it and carry it to a different spot. We have things like in our neurons, for example. So some of our neurons can be a couple feet long, a single cell. So we have motor neurons that are the body of that neuron would be in our spinal cord, right? But it has to get its axon all the way down to our foot to contract that muscle. Um, and it has to get neurotransmitters all the way down there that are made way up in the spinal cord, right? So it has to carry that stuff all the way down a couple feet. It can actually take about 2 weeks to get from our spinal cord down to our foot. But it's just constant traffic like that. Always things just moving back and forth, and these microtubules are helping with that. Yeah, go ahead. Walk along these ones in particular, the microtubules. Yeah. Yeah, good question. Yeah, so okay, so let's just take a look here, and you guys can kind of answer these. Yeah, we actually don't need to do this because I just answered it. But which cytoskeleton filaments above are involved in moving the vesicles of proteins in the video you just watched? We can open it up on the clicker just so you can put it in, but you can just check with each other, make sure you all got it. For our first clicker question for the day, just put that letter in, check with each other on your team. Should be quick because I just told you. All right, I can show you this a little bit easier now, not using my iPad. So we got 43 of the 48 of you responded today. Anybody else out there? 44? We're close. Mostly everybody's saying A, which is the correct answer. 44.
Oh, yeah.
Good. Okay. Everybody's saying A now. All right. Good. Okay. Here. Okay, good. So any questions on those parts of the cytoskeleton of the cell? Go ahead. Yeah.
Like for the intermediate components,
Yeah.
are they in the middle on the left?
Yeah, the intermediate ones here. Let's see. Oops, wrong one. Yeah, so the same one here is the intermediate. So yeah, that would be this one here.
In like the stain,
Oh, you're talking about this up here?
or like, well, can it be seen on the mouth?
Oh, there. Yeah, yeah. I see what you're saying. I believe it's the yeah, that's a good question. Which one is it? Yeah, I don't know if you can see it that well there. I think that's why it has a different image, kind of stained a different way. I think you can really only see the green and the red ones, which are the other two. But yeah, I don't think you can see the intermediate ones that well there. Yeah, good question. Yeah, go ahead.
Just
Yeah.
to be clear, the cytoskeletons only walk along the microtubules, not the intermediate filaments or the actin filaments.
The microtubules are the main ones used for that kind of transportation system. Yeah.
But then,
Mhm.
because the other ones are not.
The other ones are more for structure and support of the cell. Yeah. Yeah. Okay, other questions? Yeah, go ahead. Why A is the answer? Yeah, because that one is the microtubules here, which are the ones that form these tracks for the vesicles to move around on, like in the video we were just watching. Yeah. Yeah, good question. Okay, anybody else? Okay. All right, so we'll talk about a few more things that are just kind of related to the cell, other things that can be kind of sticking out of the cytoskeleton of the cell on the outside of the cell. One example of that is cilia. Okay, we're going to talk about cilia with different types of tissues coming up here soon when we get to the histology section in the book, which looks at different tissues. And so cilia are these little whip-like extensions that are sticking off the edges of our cells. And so, for example, we would have these inside of our respiratory tract, okay? And they're usually embedded in a bunch of mucus too. And so when we inhale a big particle of dirt or something, it can get caught in that mucus. And then the cilia kind of beat it kind of looks like a random direction if you're looking at it, but they're all beating in kind of a very organized fashion to move that dust back up. So then we can kind of cough it up and spit it out or swallow it into our digestive tract so we can get rid of it, right? So it doesn't embed itself into our respiratory tract. So that's really the function of the cilia, is to move the particles across the surface of the cell. When we're specifically talking about our respiratory tract, we're trying to get things like out of the respiratory tract back up towards our mouth. So yeah. Okay. All right, another thing that we find embedded in cells, a couple other things here, would be the flagella of a cell. And so most cells don't have these, but some do. One example is a sperm cell. So it's a little extension, but it's just one extension. It's much bigger, much more powerful than the cilia that we were just looking at. And this is for movement, right? For cells like sperm that need to move and swim through solution. We also, other cells would have microvilli, which are not quite like hair-like projections like the cilia, but more finger-like projections. And these are going to be covered with a bunch of proteins embedded inside of them. If we're talking specifically about our gastrointestinal tract, our small intestine has a bunch of these. This is what makes up what's called our brush border. And so we have brush border enzymes that are embedded into all of these. And they're the ones that do the job of kind of like final digestion of nutrients before we can absorb those into our bloodstream. Okay, sometimes when those little question marks show up like that, it's supposed to be an arrow pointing to the right. Just so you know, it just didn't render properly. But so it's an example of the microvilli, and we'll talk about this more next semester when we get to the gastrointestinal tract. Okay, so now we're going to kind of shift gears and talk about diffusion. Okay, talk about diffusion and osmosis here. And so diffusion is defined as the tendency of molecules or ions to move from an area of higher concentration to an area of lower. Okay, and this is, we're typically going to be talking about diffusion occurring across a membrane. Okay, across typically a semi-permeable membrane. So a membrane that might allow certain things to pass through and not allow other things to pass through. And so a cell membrane is very, very thin, right? It's about 1/1000th the thickness of a piece of paper, okay? And so this movement can happen super rapidly across a membrane if that membrane is open to that molecule or ion to pass through. Okay, so we'll kind of talk about different types of diffusion next, but the whole idea here, the main thing to remember is that we're going from higher concentration to lower. That's the key part. And it's really just that those particles are all moving around randomly. And since there's a lot of them in one place and not many in the other, the random movement ends up being such that more of it goes from high to low than vice versa, just because of the random movements. Okay, so the types of diffusion that we'll talk about, number one is just called simple diffusion. This one is when whatever the particle is that we're talking about is free to get through a cell membrane. Okay, so that could be like oxygen and CO2, for example. The cell membrane lets those go through no problem. Okay, certain vitamins, certain steroid hormones can just go right through our membranes of our cells. Okay, and so it's simple diffusion. It doesn't need any help. It's going to go from high concentration to low concentration. But then other things are too big or they have a certain charge to them that doesn't allow them to get through the membrane. And then we need some help. So that would be different forms of facilitated diffusion. Okay, so one of those is called carrier mediated, where there's a protein kind of in the membrane. It kind of grabs onto that thing and then shuttles it through the membrane. Okay, or it could just be an open channel that only allows certain things to get through. So for example, in our neurons, we have sodium channels. We have sodium leak channels that just are always open and they only let sodium through. All right. Same thing with potassium. We have potassium leak channels. We have little water leak channels as well. Only lets those things pass through. Somebody's very excited out there. But, okay, so there's kind of three different types there, right? So simple diffusion, carrier mediated, channel mediated diffusion, okay? But then sometimes in the cell, we move things against its concentration gradient. When we move things against this concentration gradient, it's called active transport instead of diffusion. And that would be when we move molecules against their concentration gradient, which means from lower concentration to higher. Okay. Whenever a cell does this or the body does this, it requires energy. If we just let it happen naturally, it's going to go from higher to lower, and that's diffusion. But if we're trying to do the opposite, if we already have a lot more on one side of the membrane, we're still trying to push more over there, then we got to use some energy for that. Okay. And there's two types of energy usage that can be used for active transport. The first one is ATP. So ATP, adenosine triphosphate, is a chemical currency of energy that we find all throughout the body. It drives all the reactions that require energy. And so that's called primary active transport if we use ATP to push something against its gradient. Okay. But then we also have secondary active transport, which is this one. Secondary active transport uses the energy derived from the concentration gradient of something else. Okay, so for example, there's always a lot of sodium outside of our cells, not much inside. So that sodium wants to rush in, and we can kind of use the power in that to pull other things in with it that don't have a gradient. Okay, and that's pretty much secondary active transport. Okay, so this image has those 5 different types of diffusion or active transport represented here. So I want you guys just to identify which one is which, and then on your clicker, send in the one there that's bolded and underlined, which one is secondary active transport. Okay, so talk with your team, figure that out, and send it in when you're ready.
Oh.
Yeah, like a 10 outside those.
Question.
Sure.
I thought there were more than 5 flagella per like certain cells. Is that?
Not typically in human cells, maybe in some microorganisms there might be.
Okay,
Yeah.
that makes sense.
Yeah.
Thank you.
Yeah.
I just took a microbiome class, so.
Yeah, totally. Yeah, I'm sure there's some bacteria and stuff that probably do, but yeah.
You are right. This might be the energy.
Secondary.
That's not me.
So it's been really. This carrier media is. There's another spot and it's the plastic. Well, that's what I heard.
All right, make sure you're on the same page as your teammates and then send that in when you're ready. You're just sending in the one that is secondary active transport. All right, about 6 more or so of you need to send it in. Just hit that button again just to make sure. So you guys are pretty well split between C and D. About 40% of you are saying C, and then about 55% saying D. So just within your team, talk about the difference between these two. Talk about the difference between these two, what makes them different, and change your answer if you need to.
That is the energy for ingredient.
I'm just.
Oh, and it's secondary to spin too? But what's the secondary?
Secondary.
Oh, there's two.
Sure.
It doesn't use ATP at all. Then that answer my question.
That answers your question. Answers the question. Yeah.
Is the reason between the difference between a channel and carrier medium, is it because some molecules are too big, so they can't do a channel that's like too big to let everything in?
Yeah, that's definitely part of the reason for sure. Yeah, yeah.
So like it's also be like too big, so they'll use carrier medium.
Yeah, if it was an open, big enough channel for that, then it would just allow other things in too, probably.
So
So,
channels are usually like ions and stuff.
typically, typically smaller things and charged things, a lot of times, so yeah, ions, yeah. Yeah, yes, good question. All right, go ahead and put your final answer in there if you haven't yet. Man, you guys didn't do a good job of convincing each other after I had you look at the difference between C and D. Oh, now you're starting to shift. Okay, all right. Well, we've got about 2/3, maybe 60% of you saying D, and then the other 40% saying C. Okay, so if we go back to the definition of primary versus secondary active transport, primary uses what? For energy. Yeah, we need energy for active transport. Primary active transport uses ATP. Okay. The key feature with these is both of the things here, there's sodium, okay, and there's potassium. Both of them are moving from low to high concentration, right? The opposite of diffusion, right? There, so that requires energy, right? The purple ones, the sodiums, there's not much sodium over here, right? There's already a lot over here, but we're pushing it that way, right? With potassium, there's not much over here. There's a lot in here, but we're pushing it that way. So both of those are going against their gradient, right? So that's the key. And the fact that you see ATP there is the other key, that's primary active transport. This one, glucose is going into the cell and sodium is going into the cell. There's already a lot of sodium out here, but not much down here, right? So sodium's going with its gradient. It's going from high to low, but glucose is the opposite. There's not much glucose out here, but there's a lot down here, but we're pushing more of that glucose in. So it's kind of like sodium. It's kind of like the secondary active transport is kind of like a teeter-totter type thing, right? So it's like glucose is here. Sodium's gonna sodium. We got the power in sodium trying to rush across the membrane. It hits the teeter-totter and flings the glucose in with it, kind of, right? So we're like using the energy from that sodium to kind of fling the glucose in, and so that's secondary active transport, right? No ATP needed. We can harness the energy in another molecule or ions concentration gradient to pull another one with it. Okay, questions on that one? So D is the right answer for the secondary active transport. This is secondary, this is primary. Okay, what's A? Up there. That one's the channel mediated, right? So it's channel mediated. What's this one? Simple diffusion, yeah. Things that can just get right through the membrane without any help. And then this one, carrier mediated, yeah. So it's a little bit, it's not just an open channel. It's kind of a little bit more of a mechanism where you grab onto it and kind of push it through instead of just having an open channel to let it through. Yeah. A is the channel mediated. B is the simple diffusion. Okay. Questions on those? We're good? Okay. All right. Well, then we're going to continue on with the next part. If I can get this to move. Hold on a sec. There we go. Okay. All right. So our next kind of homeostatic mechanism, we talked about pH last time or on the first day, right? Osmosis is the other part. Okay. And so diffusion, we already talked about, is that movement of substances from higher concentration to lower. Osmosis is diffusion of a solvent. Really, we're going to be talking about water. Okay. It's water moving through a selectively permeable membrane. Okay. And osmosis is going to be moving towards the space where there's more solute. Okay. So we'll keep talking about that and what that means in a second. So there's different ways that water can get through a membrane. It can get through a membrane just because the wiggly lipid layer of the membrane, kind of when it wiggles around, it opens up a little bit and water can kind of squeeze through. Okay. But then we also have specific protein channels that just allow water molecules through. Those are called aquaporins. Okay. And so a real key definition here is osmolarity. Okay. So the osmolarity of a solution is the concentration. That's what those little brackets mean, right? Concentration of all the solute particles in a solution. Okay, so the more particles, the more solute we have in a solution, then the higher the osmolarity of that solution is. The less particles we have, the lower the osmolarity is. Okay. Okay. So that's our key definition. And so then how does this apply to osmosis? Okay. So the idea of osmotic pressure is that the water has the tendency to move towards the space that has higher osmolarity. Okay, so space has higher osmolarity if it has a higher solute particle, meaning like more stuff dissolved in the water. So more ions, more other substances, okay, that creates osmotic pressure. And so we typically think of pressure as something that pushes like away from itself, right? Like pressure in your blood was like pushing out on the blood vessels, blood pressure, right? Osmotic pressure is the opposite. The higher the osmotic pressure is, the more it pulls water towards it. Okay, so high osmotic pressure is due to more particles or more solute relative to the fluid, the amount of fluid that's there. And then low osmotic pressure would be due to less particles relative to the amount of fluid that's there. And this is always, we use these terms kind of relatively because we're talking about a cell membrane. And then we got one space on one side of the cell membrane, usually inside the cell, versus the other side of the cell membrane, which is outside the cell. And so if we say the inside has high osmotic pressure, that just means it has higher osmotic pressure than the outside. Okay, it's just relative to the other side of that membrane. Okay, so high osmotic pressure means that it's going to pull fluid towards it. If a solution has low osmotic pressure, then it doesn't have the ability to hold on to that fluid, so it lets it go. And fluid's going to go away from that low osmotic pressure. Okay, so if a fluid has high osmotic pressure, you can just write in here, it also has high osmolarity. We could also call it hyperosmotic. This is where I need my iPad, but I can't write that right now. Hyperosmotic, high osmolarity, high osmotic pressure, all those things would go together. Okay, if it's low osmotic pressure, then it's got low osmolarity, and it's hypoosmotic. And again, that's just comparing it to the space on the other side of the membrane next to it. Okay, so the classic example is right here, right? So we got this semi-permeable membrane, we got fluid on both sides, we got some particles on both sides, okay? And so what you want to look at is the purple balls are the solute, right? The pink solution behind it is water. And so look at this and answer the questions. Answer all those questions, and then the one on the bottom is the one I want you to answer on your clickers, but do that with your teammates here. And yeah, answer that question on the bottom on the clickers, but answer all of them in the line there first, okay? Go ahead.
The ratio of water.
Breaking your chair.
He said he's.
Oh, that one was definitely broken, yeah.
I'm like a chronic chair tipper. These are just like the worst.
Can't tip them, yeah.
Cause it's like, I just.
He says he's snapping.
I already snapped.
Really? I've seen those broken ones around classrooms, yeah.
Stand up and person bodies and like that.
Did you go to the Savannah Bananas over the weekend?
My mom did.
Your mom did?
She gave me a shirt.
Okay, nice. A couple of our kids went with friends. They said it was a blast, yeah.
It's a friends of mine, like 14-hour shirt.
Oh,
Like,
yeah, that's a lot. All right, send in the answer there on the bottom. Which way will water go? Will it go to the right from A to B, or will it go to the left from B to A? All right, about 5 more of you, send it in. All right, I'll leave it open for a sec. A couple more of you still need to send it, but it looks like most of you are saying B. So about 90% of you are saying B. So that means it's going to go to the right. It's going to go from A to B. Okay, let's just go through these questions quick here. So which side has more solute? Which one? B, right? Which side has higher osmotic pressure? B. Which side is hyperosmotic? Which side's hypoosmotic? Good. And so then water is going to go towards the side with higher osmotic pressure to the right. right? And it'll look something like this, right? When water moves, it's going to shift over that way, right? So the water molecules followed that higher osmotic pressure and went to the right. Okay, so that's osmosis, right? Osmosis happened and it moved to the right. So let me just do let's do one kind of practical example of this, and then we'll do all those slides with the red blood cells at the beginning of class next time. But one practical example of this is when someone sprains their ankle. Look right there. Yeah. All right. Some of you have done that before. Doesn't feel good. But when you sprain your ankle, your ligaments tear. And when that happens, you tear other tissues too. And some cells break open, some blood vessels break open, and all the stuff that's inside of your blood vessels and your cells, it all spills into the space around your cells. Okay. So you've got three spaces in the body. You've got inside your blood vessels, you got inside your cells, and then you got this third space, which is where swelling happens, called the interstitial space. Okay. So when you get an injury and you break all this stuff open, you break cells open, all the stuff that was inside of those things is now just kind of flooding that interstitial space, right? And so that's ions, that's proteins, that's all kinds of things. All of those things create osmotic pressure, right? So now all of a sudden we just added a bunch of solute to the space around our cells that wasn't there before. So our osmotic pressure in that area is way higher than it was before, right? So we end up looking like this, right? Because all the water, a lot of the water from our blood vessels and from our cells surrounding that area flows out, right? So these cells are actually going to shrink. These blood vessels are going to shrink down a little bit because they lose fluid, but all that space is now filling with fluid because of the high osmotic pressure there. And that's why you get the swelling stuck there, right? You can massage the swelling and try to push it out, and it will kind of push out, but then it just comes right back in a couple of minutes. How many people have dealt with that with an ankle sprain? Yeah, a few of you. Yeah, it just comes right back because you're not pushing all the proteins and stuff out. They're all just stuck out there, right? You got to wait for your lymphatic system and for your immune system to clean all that up. But that's an example of osmotic pressure. Okay, we are out of time, but we'll start off with this little review stuff next time. Okay, have a great rest of your day, guys. We'll see you on Wednesday.
Yeah.
I do. For lab.
Yeah.
For a lab quiz, is it gonna be on the so this last lab that quiz that we
Yeah.
took was on our packet, but we kind of actually discussed it.
Yeah, it's not like that.
Be on our phone.